Hello, everyone, and welcome to today's webinar, IR Biotyper, New Insights into Microorganism Strain and Serotyping by FTIR Spectroscopy. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Brooker. To learn more, visit brooker.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Dr. Markus Meyer, Product Manager, IR Biotyper and Care Consumables at Brooker Daltonics, and Dr. Maria Pardos de la Gandara, Deputy Director of the French National Reference Center for E. coli, Shigella, and Salmonella, Institut Pasteur. Markus and Maria, you may now begin your presentation.
Thank you very much for the kind introduction, and welcome to everybody here for the webinar on the IR Biotyper, giving some new insights into microorganism strain and serotyping by FTIR spectroscopy. My name is Markus Meyer. I'm the Global Product Manager for the IR Biotyper. And I would like also to kindly introduce Maria Pardos de la Gandara. She will give the second part of the presentation with results from the Institut Pasteur. Let's get started. So I will give you a very brief introduction into FTIR and IR Biotyper product description, and then I will turn to some comparison of IR Biotyper and multi-biotyper. Maybe you know this product, and give you also some insights into our news and an outlook in the future. And then I summarize and will pass over to Maria. So first of all, a quick introduction and some questions. So there are some open questions and needs in hospital hygiene and infection control. And one of them is, of course, how can we analyze outbreaks? How can this be performed? And is, for instance, this an outbreak strain? Is it one cluster? Or is it something different? Do we have really seen this strain, or is it something new? And one important question in addition is, how can we faster and better monitor the distribution and the transmission of pathogens more or less in real time? One important question that is following then is, how can we prevent outbreaks, and how can we implement screening approaches to be faster and more preventive? And there are some specific questions, for instance, differentiation of serogroups, serotypes for distinct species. For instance, it could be pneumococci. You will hear a little bit about Salmonella, Legionella, Listeria. Also, these are some questions that will be answered. FTIR principles and some definitions. I make it really short. So it's all about IR. It's infrared light. It's an electromagnetic wave. I think you know this from your time, from your study time or school time. That's an electromagnetic radiation, which is defined by the wavelengths, linear frequency. What you will also hear in the next slide is the wave number. Wave number is defined as the waves per centimeter. And what's about FT? That's Fourier transform. That's a kind of quite complicated mathematical operation, which is increasing the speed and analysis. What are we using here? We are using, Bruker is using in this product absorption IR technology. So there are different ways how light, infrared light can be used. Maybe you have heard about fluorescence, transmission, scattering, or light can be reflected. But here we're using absorption of the infrared light. And what is the absorption about? The principle here is that we take a kind of a snapshot of an intact biomolecules in the cell. As you know, the structure of a typical prokaryotic cell made of course, water, salts, and so on, but there are three major, very important biomolecules. These are the carbohydrates, many of them on the outside surface of the bacteria, and lipids and proteins, and of course other molecules. But these are the three important ones. And if we look what FTIR spectroscopy is doing in terms of the absorbance, you see on the one scale you see the absorbance, on the other scale you see the wave number, and there are different regions in the spectrum where different biomolecules show some absorption. There's on the left-hand side, there's the so-called CH stretching for fatty acids. And more in the middle, there's the absorbance for proteins, peptides, and on the right-hand side, you see the so-called CO stretching for polysaccharides, which are very important for many differentiating steps if you look on bacteria. So what is about these further basics and processing of spectra? First of all, the selectivity of this technology is very high, especially if you look on the carbohydrate region, which is very important. And this allows the differentiation of quite closely related microorganisms. And this means strain typing. Maybe you know strain typing with other technologies like PFGE, so some electrophoresis technologies or some PCR technologies. Here we are using the complete cells and the absorption to differentiate the strains. And if you look on the spectra, it looks not very differentiating the raw spectra. Here are 5 different strains with several replicates. It looks quite similar, but after some mathematical calculation here, the second derivative of the specific wave number region again is taken, and here you see then the differences between the different strains. Different colors here are the different stains, and you see the replicates, they fall together, but you see differences between the different strains. So also here it's some mathematical calculation that is needed. All of this is combined in a real workflow. We call it the Biotyper workflow, and this complete workflow takes roughly 3 hours for roughly 30 samples. The workflow starts with harvesting. So we need grown cultures, a pure culture, and this culture is taken into a tube where suspension, so specific metal cylinders are incorporated, and this is needed to get a good suspension of the bacterial cells in an ethanolic solution. Then this solution or this suspension is taken and pipetted on the target plate. This is a standard 96 spot plate where the infrared light can be used for, and then it's simply dried on this target plate, and it's then inserted into the instrument. And roughly 1 minute per spot is the time. So this means 1.5 hours for the pure measurement, and for the other sample prep steps, another 30 to 1 hour. And then the most interesting step starts, of course, because the bioinformatic analysis, that's a very important step, and where most of our energy is also going to optimize this for our customers. So we think the IR Biotyper is a complete solution, complete strain typing solution. It's made of a hardware system, a plate reader, spectrometer, and this is optimized for the microbial analysis. And we are based here, or this technology is based on a quite huge experience from Bruker Optics leading technology in terms of IR spectroscopy. It comes with a software that was developed by Bruker Daltonic's microbiology team. And this means that we have algorithms and visualization tools that are really optimized for the microbiologist lab. We provide some standardization here with the kit solution. So the kit is providing some standards, process controls, lyophilized bacterial cells where differentiation must be seen, but the strains are very close to each other. And the suspension vials, which I explained in the slide before, are coming also with the kit to generate really and provide some standardization here. What's now about the differentiation or the difference between multi-biotyper? I think you have heard about this product, and subtyping, and the IR Biotyper. What are these different products doing? So multi-biotyper and subtyping, this is based on multi-MS, multi-TOF, time of flight, mass spectrometry. And this is using peptide and protein fingerprints, a very stable and robust technology, and nowadays the gold standard in microorganism identification on the species level. It comes with a large database of roughly 4,000 species, and the subtyping module means this allows some subspecies differentiation, for instance, for very closely related species like Listeria, and some interspecies differentiation is also possible to detect some resistant markers, for instance, for Bacteroides fragilis or for Klebsiella. What is the IR Biotyper in contrast doing, or in combination? This will come later. It's more based on cell phenotyping. You have intact cells here, and you have the infrared technology, which I described. This is a strain typing method, so this allows differentiation more on the isolate level when the identification of the species is already known. And it can be used for analysis of outbreaks in real time, for hygiene and infection control, for prevention and fast screening. And it comes also with first databases of so-called classifiers. I will explain a little bit later. And this can be used for serotyping, for serogrouping, for instance, for Streptococcus, for Legionella. And there are first serogrouping O serogrouping classifiers for Salmonella and also for Listeria. Coming to the new features, the news of the IR Biotyper solution, and a little bit also the outlook. What is our focus to improve further the product and make it interesting? So first of all, I explained the multi-biotyper and the IR Biotyper. These instruments and these solutions, the workflows can be combined now. So MBT, multi-biotyper can be combined with the IR Biotyper in terms of software and LIMS system. We have largely improved the GUI to make it more easy for our microbiology customers. The usability, now it comes with an easy reporting, PDF report, especially for larger data sets. The performance was improved. It's easier to handle. And this is one important thing here, the machine learning, the classifiers, which I already mentioned. This is very important because this is a learning system where you can put in known samples and can detect unknown samples by ready-made classifiers. We have extended the wave number region so that we can cover also the lipids, proteins. The carbohydrate region still remains very important. Most of the differences are in this region. And there are some new, very nice visualization tools really helping our customers, for instance, like a 3D scatter plot and other tools to visualize and display your data. So multi-biotyper, IR Biotyper working together. So this is more or less the slogan here, and it shows that microbial identification, species level, and strain typing, they're coming closer together as one solution. And I think that's a very important thing here to show. What is about these classifiers? That's a quite new software tool that we have in the software since roughly 1 year. So that offers really a new and fast way to look on real-time strain typing. And what is done here? So we start with training sets of known strains, known isolates, serotypes or serogroups, and they are built into a so-called statistical model. And this supervised model can be used for classification of unknown samples in real time by comparison to the known strains to this model. And this is based on artificial neural networks. It's a machine learning algorithm. This is a quite innovative bioinformatics tool to generate these classifiers. And if we summarize this, there's an input, it's a training data set, known samples, and then the algorithm learns to recognize these known samples. And as an output, you can classify unknown samples, for instance, for pneumococci zero grouping, for Salmonella, for Legionella and Listeria. And with this, I would like to summarize and conclude. So I hope I've shown that FTIR provides high discrimination power based on phenotyping of intact cells. The IR Biotyper is using FTIR as a fast technique. Latest software developments provide user-friendly tools to quickly analyze outbreaks and detect also serogroups, serotypes by ANN, and the connection between the gold standard for microbial identification, the MALDI Biotyper, with the IR Biotyper opens new dimensions for full characterization of species and strains. So that's quite interesting. And with this, I want to thank you, particularly the collaboration partners. A special thank to Dr. Maria Pardos de la Gandara, and of course also the Bruker team, here R&D team, the marketing team, technical and application support. So they are very important to keep the product here and to make it really a value for our customers. And with this, I want to thank you, of course, for your attention. I'm happy to get later the questions and would like to pass over to Maria now.
Thank you, Markus. So, as it has been said, my name is Maria Pardos de la Gandara. I'm the Deputy Director of the French National Reference Center for E. coli, Shigella, and mainly for Salmonella in the Institut Pasteur in Paris, here in France. The Enteric Bacterial Pathogens Unit is a research unit in Institut Pasteur that hosts two important centers regarding Salmonella. First, the French National Reference Center, in which we monitor with our main activity is to keep the surveillance of Salmonella circulating among humans in France. We receive about 10,000 samples every year, specimens. We track the emergence of new serotypes and new resistance profiles in these Salmonella isolates, and we monitor the increase of number of cases of human salmonellosis. And very important also, we have to report immediately to the Department of Health any case of typhoid or paratyphoid fever that we identify, as well as any collective food poisoning, which is when there are two or more patients with similar symptoms because of eating suspiciously the same food in a sort of same period of time, more or less. Another center that is important in this unit is the Collaborating Center for the World Health Organization on Salmonella, where we provide our expertise in typing as well as in resistance to antimicrobial. So just a little introduction, because Salmonella has a complicated taxonomy. We are talking about a gram-negative rod, which can be anaerobic, and it is mobile thanks to flagella. Salmonella is the genus, is the name of the genus that belongs to the Enterobacteriaceae family, and it is divided in two species. Salmonella enterica, which is the main species affecting humans, and Salmonella bongori, which can affect some animals, but it's really rare to be found affecting humans. Salmonella enterica is also subdivided in 6 subspecies by the classical methods, and there are 2 new subspecies called by now 7 and 8 that have only been identified when using whole genome sequencing. Then all Salmonella enterica and bongori are divided into serotypes, and we have at the moment up to more than 2,500 different serotypes. Some of them, as you can see, have their own names. And well, to summarize, we never talk about Salmonella enterica subspecies, enterica serotype Kentucky, for example. We would directly say Salmonella typhi, and this is how I will talk about it today. Otherwise, I will take forever. Salmonella resides in the intestines of infected animals. We say it's a zooanthroponosis because humans will get infected when eating contaminated food or through very close contact with animals. And it's not only about professionals working in farms. We also have to think sometimes about pets, and not only warm-blooded animals, but as it starts to be more frequent than before, cold-blooded animals, such as reptiles. And in some cases, very important, but very rare in countries where it is not endemic, the human-to-human contact, like the first historic case that was Typhoid Mary, and that I invite you to investigate further later on. Salmonella, for most of serotypes, which we call minor serotypes or non-typhoidal serotypes, causes a self-limited gastroenteritis. but there are these few major serotypes, important ones, typhi, paratyphi A, paratyphi B, and paratyphi C, which may cause a systemic infection called typhoid or paratyphoid fever, which can be potentially lethal. Okay, so traditionally Salmonella has been classified using a first step of biochemical identification following the subspecies regarding a set of substrates, and followed by the serotyping, in which an agglutination must be done on plates using over 160 antibodies and following the White-Kaufman-Laminar scheme, which has been the gold standard to identify serotypes for over 90 years. You can see on the top right an extract of this scheme, and the process is usually first identifying what is called the somatic antigen, that is, we find by agglutination the epitope of the lipopolysaccharide in the wall of the cell, and then we follow with the identification of flagellar antigens, usually two phases, phase 1 and phase 2. And with these three antigens, somatic, also called O, and the flagellar, H, 1 and 2, we may have the traditional serotype, the serotype profile. However, since 2018, we have moved in the National Center of Reference for Salmonella here in France to whole genome sequencing as the first intention method for typing. So instead of serotyping, the lipopolysaccharide and the flagellins, we are performing MLST. I don't know if you will know about it, but I can take some questions about this later on if needed, which is based on this multi-locus sequence typing, 7 genes that are regarded, and the 2 genes encoding for the flagellin. Whole genome sequencing can also allow us to do some subtyping that in our case we do based on the 4 genome MLST based on 3,000 genes. And finally, even if needed, we can do some clustering, really detailed clustering based on single nucleotide polymorphism. So these new methods are giving us a lot of information. We can know a lot of things from the bacteria, including all the acquired genes for resistance to antibiotics. However, it requires a long processing compared to the serotyping, and it is unable to respond to urgent needs. As you can see in the graphs in the bottom, we have lost the ability to give fast answers for serotyping that we had in about 6 to 10 days. This is gone, and the fastest we can go with the sequencing at the moment is about 15 days. So this is when we got interested about this infrared biotyper, as Markus and other colleagues are telling us the idea that it can be really fast in detecting serotypes, serogroups at least, and probably outbreaks. So we set the test. And the first thing we wanted to do was a proof of concept and see if it really could separate strains belonging to different O groups. That means somatic, so lipopolysaccharides based on sugars. And for this, we just took a small set of samples. We took 10 strains, passed in triplicates, each one of them, some of them belonging to the typhi serotype, so O9, and some of them belonging to both Typhimurium and paratyphi B, which are O4. And we run the basics with the IR Biotyper. As you know, I'm not a bioinformatician. I'm a microbiologist, a clinical microbiologist as formation. So just what the machine can give me without any manipulation. And we were very happy to see already in this dendrogram application that it has how Typhi, let's say the O9 group, was well separated from the O4 serotype. The same strains passed by the TCA analysis. So on the top right, you can see the average spectra for each triplicate. It generates one average spectrum, and in the bottom you can see the 3 triplicates separated and how well the system managed to separate the O9, the Typhi, to the right in different tones of red, and from the O4s that are on the left. This is the same analysis, just shown in these magnificent 3D representations that I personally like a lot. Again, with the Typhis, the O9 is very well to the right. And the new feature, the LDA, that seeing it in 2D doesn't add a lot of information, but if we go to the 3D, we can see also the yellow buttons representing Typhimurium separated from the different paratyphides in green and blue, and again, the typhus B09 still aside. Okay, these were great results, so we went a bit further and we said, let's try to differentiate serotypes that share the same group O, the O4, same glosses, but for which we know that the complete serotype is different, the complete antigenic formula is different. And for this, we took more samples of Typhimurium and paratyphi B, the real para B, which is a major serotype, and the paratyphi B biotype Java, that is considered minor, it's a biotype of paratyphi B that causes gastroenteritis instead of paratyphoid fever, so clinically very important. This time, a bigger set of samples, so 55, and still in triplicate. And again, we have quite some promising results because already with PCA, Typhimurium is mostly separated to the left. You can see the yellow dots or balls in the 3D representation, and how the paratyphi B biotype Java, which are the green squares, seem to cluster somehow together compared to the paratyphi B, the real paratyphi Bs that are outnumbering all the other two. And quite similar results with the LDA. We, of course, will need to go a bit further and analyze these two outliers from Typhimurium that didn't want to cluster together with all the rest, but as preliminary results were quite promising. So why not to go a bit further? And we try to compare these two biotypes, the real paratyphi B causing the paratyphoid fever, which is quite homogeneous. It's usually related only to MLSP 86, and compare it to the biotype Java, which we call it as one thing, but it's actually quite heterogeneous and it includes many different STs. So we took several of them from our collection and we ran a comparison. And well, we need to go further in this analysis. This has been just done in 10th of August and we need to analyze further. But especially looking at the LDA, it looks quite promising. It could look like paratyphi B are quite grouped in the center and how the different SPs of the biotype Java are a bit more scattered around. Something that Markus has mentioned and we also wanted to try are these classifiers, this machine learning. So we did use the Bruker classifier, the supervised model that contains a well-curated collection of different Salmonella serogroups, and we run it in our isolates. In this sample, in total of 65 isolates, meaning 195 spectra, because, well, it runs on every single spectrum, as you can see on the right. And it was quite successful, it was successful. We found the right group for over 93% of the samples. You can see how the system will give you the O, and you can see it in a column called project on the right. And the machine itself will give you a confidence that will be differentiated by colors. So green when it's very sure of itself and red when it's not so. It worked very well. However, there were some, again, outliers, like that little Typhimurium in the bottom that went to the O group 8. Discussed with the technicians, there are things that we need to figure out because it could be a problem of the sample, not just quality, but if they are rough strains that would affect to the oligopolysaccharide of the strain, and then it would explain why the classifier got it wrong. The conditions of the preparation are important. Which culture media was used? Well, we know that the classifier by Bruker has been optimized for cultures on blood agar, and we did run things both in Miller-Hinton and TSA plates. So we might have to work on that. And there are some conditions that can influence the results, like humidity, which was quite variable in our laboratory those days that we ran the samples. And also related to this classifier, we wanted to test the reproducibility of the system. So we just ran one same plate on a Friday, and the same plate just stored at room temperature was run Monday, so 3 days later. And it we were happy to see how a similar deviation was observed in all the spectra. You can really visualize, especially on the 3D representation, the plate of Friday and the plate of Monday, how every isolate has a comparable deviation. And even better, that when we run the LDA, which corrects the effect of humidity, the 6 now copies of the same isolate really clustered together. This means that all the spectra stored by the machine in different experiments are comparable, so you can create your own database from which you can end up creating or enriching your own classifier. So which are the next steps for us? First of all, we want to see how well the infrared biotyper can identify outbreaks. So we will compare some sporadic strains from clusters that we know are epidemiologically related, and to see how well the differentiation is done. And on the other hand, taking advantage of the vast collection of different serotypes gathered by the WHO Collaborating Center, we want to test how well the different serotypes, at least the O groups, are differentiated. So to summarize, from our part, the infrared biotyper was really well able to differentiate Salmonella strains from the group 9, in this case typhi, versus group 04, represented by some strains from Typhimurium and Paratyphi B serotypes. It was impressive how the LDA was able to correct the effect of some acquisition conditions, mainly the humidity, and how this correction allows really to compare the spectra from different runs. So as I said, you can create your own database, you can improve your own classifiers, so it will be performing better and better every time you use it. We hope so. Our further work anyway will need to analyze those sublayers that I showed you here and there, and we really want to know how Biotyper can discriminate outbreaks from sporadic isolates. We are really thrilled about this part. And we want to test to the limit this classifier ability to identify different serotypes, or at least strains from different O groups. So with this, I just want to thank you for your attention. Again, to thank you for being invited in here. And we are very ready to get any questions or comments from you. Thank you.
Thank you, Markus and Maria, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how many samples can be measured by the IR Biotyper per day?
Yeah, that's a question for me, for Markus, I think. As I've explained in my talk, we have roughly 30 samples in 1.5 hours measured. So this means in principle you could measure more than 100 samples a day, or let's say 150. Depends a little bit on your workflow in the lab, but the principle of the measurements, you can do a lot of samples per day. Let's say several hundreds.
All right, thank you. Next question. What is the difference between MALDI Biotyper and IR Biotyper?
Also, this I touched in the presentation. The main difference is here that the MALDI Biotyper is giving you the species identification by MALDI TOF MS, gold standard in identification of microorganisms, and the IR Biotyper is a tool for strain typing to look further on differences, for instance, based on the carbohydrate or different other biomolecules. But we really try to make it quite clear. So species identification is MALDI Biotyper, that's the gold standard. And if you need to look further, the IR Biotyper is a tool for further differentiation, to get further differences, for instance, on the isolate level.
Thank you. Next question. Are customers able to build their own classifiers?
Yeah, that's possible, and the software is open for this, and this is really also our intention at Bruker, that because the differences and the local questions, especially for hygiene and infection, are sometimes so different that we cannot have everything in our facility to build those classifiers. So therefore, we keep it open, and the customer himself, they can use the classifiers, but also the other tools that are in the software to build their own database. Yeah, that's possible, to make it short.
Great, thank you. Next question. What kind of classifiers are included in the IR Biotyper software?
Yeah, I think I mentioned also some examples in the presentation. So first of all, we have I think quite big classifier for Streptococcus pneumoniae, holding more than 30 different serotypes, serogroups. We have started, and this is therefore, of course, very interesting that to see in Maria's talk, that we have started to build first Salmonella O serogroup classifiers, and we have first classifiers for Legionella to differentiate the major groups. So that's serogroup 1, that is differentiated to the other 2 to 15, I think, serogroups. And we have also a first classifier on Listeria questions. So that's already in our latest software, and we are, of course, working on more classifiers which will come in the future.
Great, thank you. Next question. Can you use FTIR for species level identification?
In principle, this was, if we look a little bit in the history of species identification and strain typing, IR technology was used for species identifications. In some cases, it's possible, but we make it really clear that the species identification, that the MALDI is the gold standard there. And there's some overlap, but we see really clearly the MALDI being there really ahead of IR for pure species identification. So therefore, we try to differentiate here very, very precisely.
Great. Next question. How many Salmonella serotypes can be distinguished by the new IR Biotyper?
So that's a little bit, yeah, maybe Maria can help me a little bit, because I think that's also an open question. I think, as you know, yeah.
Yeah, no, that's part of what we want to try with our database, especially from the WHO collaborating center that has normally at least one specimen of each serotype, and we will test the maximum. Right now, the answer is we don't know. We hope as many as possible. The more it's more about differentiating more than purely identifying. You will identify if you have a good classifier or a good previous database. So first of all, what we try is to see how it differentiates different things, and then how it really identifies, which is not exactly the same intention, but it is an open question. We still don't know. We will see.
Thank you. Next question. Has the IR Biotyper been compared to whole genome sequencing? How did it perform in comparison?
Yeah, that's maybe also a question for me. There are different literatures and publications out. Of course, whole genome sequencing gives you, yeah, as the name says, the complete genome, a lot of information. So we cannot say that IR Biotyper is giving always the same results and has the data depth of whole genome sequencing. But in many cases, and you see it in several publications, especially for some gram-negative species, for instance, Klebsiella and also Escherichia coli, we see a very good overlap in some of these publications. But whole genome sequencing is giving you, of course, the complete picture. So therefore, the comparison is not always it's not a complete overlap, but in many cases we see a good overlap.
Thank you. Next question. Is the IR Biotyper supposed to replace the MBT Biotyper?
Yeah, I think this is, I've also addressed this also with some of the previous questions. No, absolutely not. So we see the multi-biotyper as the gold standard for species identification. And so therefore that's a very clear, yes, multi-biotyper will stay. That's the gold standard, and the IR Biotyper is giving an additional orthogonal technology to do further differentiation.
Okay. Looks like we have time for one more question. Can we teach the system implement the database with our own strains?
I think this is also for me, is to implement own strains in the database. I think we had a quite similar question before. I think, yeah, you can of course use your own strains. That's the intention. I'm not sure if I answered completely now. Maybe you can repeat the question one time.
Can we teach the system implement the database with our own strains?
Can repeat the system. Maybe Maria, you can help me on. I'm not sure if I got it completely.
I'm understanding this is about what we are discussing, the incrementing the database of the classifier eventually of the system. To this, the answer would be yes. So all the spectra that you read get recorded by the system, and you can choose to use them or not as a comparison for the next coming. So as I said a bit in my presentation, what we think is that the more strains you put inside, the ones you trust, the ones that were good in quality, you can use them to feed your classifier. So the bigger, the better, because it will get more accurate in future identifications.
All right. Well, thank you, Markus and Maria, for your informative presentation. We will now start. Thank you for your time today and your important research. We would also like to thank LabRoots and our sponsor Brooker for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.